Oh, where are the breakout rooms? Why did you point? So good morning, everyone. I'm happy to see you all here. My name is Mariana Carbone, and I'm the program manager for CIPHER and the PSMCC. And thanks for joining us. This is our first virtual PSMC collaborative showcase event. And just a note that there's no confidential information should be shared, and we are being recorded. And I have to give a big shout out to Dr. Deborah Thompson. This event would not be happening without her help and expertise, so I really want to thank her. And she will be moderating the questions after each speaker, so feel free to type them into the chat or raise your hand or just jump in. At the end of our speaker sessions, you'll have the opportunity to join a breakout room for some individual time with our speakers, and I'll open up those rooms at the end. So I think we're ready to start now. So before our research talks begin, we have 3 opening speakers. So first up is Dr. Stephen Lommel, the Associate Dean for Research and Director of NCARS. Please go ahead, Dr. Lommel, if you're ready. The floor is yours.
Thank you, Mariana. Good morning, everybody. It's good to see you. I see a lot of my old plant pathology colleagues in here. So hi, everybody. Hopefully we can meet in person soon. I think we're moving in that direction. Want to welcome you to NC State University and the College of Ag and Life Sciences. And we're here for our first Plant Soil Microbial Community Collaboration Showcase, PSMCC Showcase. These showcases Deborah puts on in a number of our areas in the college. My name is Steve Lommel. I'm the Associate Dean for Research here in the college and the Experiment Station Director. Just want to give you a little bit of background information on the college and the university. NC State is, if you don't know, is really a complete ecosystem for plant sciences innovation and improvement. We work on crop diversity and yields, sustainable agricultural systems, and the translation of those things into human health outcomes with our Plants for Human Health Institute in Kannapolis. North Carolina is one of the most diverse agricultural states and agricultural economies in the country. And that's evidenced by the fact that we actually have over 90 different commodity groups in the state, spanning the spectrum from animal production to row crops to many specialty crops. We also have great diversity in our different soil types and climatic conditions, since we go all the way from the hot coastal plains up into the mountains. The state has 2 land-grant universities. NC State, which we're here at today, is one of them, as well as our sister university, North Carolina A&T. They're land grants. They both have the same mission. In addition, in partnership with our Department of Agriculture, we have 18 research stations and 4 field labs scattered throughout the state, representing most of the climatic places in the state and representing many of the climatic conditions across the country. We have the North Carolina Cooperative Extension Service, which has an office in every one of 100 counties, including the Cherokee Nation. And so we have this really robust network of facilities and human talents. So if you want to interact with us, we can do field trials and deliver results within our single management system within the state and represent a large part of the nation. We also stood up the Plant Sciences Initiative recently, and hopefully you've all at least heard a little bit about that. It's predicated on 4 platforms: plant improvement, data-driven sciences, plant sciences, resilient agricultural systems, and education and outreach. And as you'll learn today, the PSMCC is integral to a number of these platforms for the PSI. So the PSMCC's goal is to promote innovation and advance and create advances in a variety of research approaches, including computational genomic and experimental tool development and applications to ultimately improve plant health and increase crop yields. Today we have a number of stakeholders together, so we're going to talk about this. And you're also going to learn how this fits the mission of the PSI. I should say that the university, in addition to the Plant Sciences Initiative, has a number of other big idea efforts going on, and we really won't talk about today's, but I'll just rattle off a few. We have the Plants for Human Health Institute, the North Carolina Food Innovation Lab, which are more involved with what happens when you eat food and prepare food for human consumption. So we sort of have the entire pipeline. We have scientific data academy that we're standing up that spans the entire university in artificial intelligence and these type of things with applications across the entire university. We have a coastal resiliency initiative. And so we have quite a few different initiatives going on in the college and the university. And to drive these initiatives and the Plant Sciences Initiative, the college has hired over 110 faculty in the last 3 years. That's a third of our college has rebooted with new faculty in the last 3 years. And I would challenge you to know of any other college that's done that much hiring in the last few years. I see many of these new hires on the Zoom call today, so you'll be hearing from them. But that's really created, I think, the engine that's going to make the Plant Sciences Initiative successful, as well as the PSMCC, with all this great young talent that's collaborative, interactive, aggressive, coming in with all the new skills and ideas. I think there's really an opportunity here to do some great stuff. And my final thought is that our key product as a university for you industry folks is our graduates, and both undergraduates and graduate students. We are training your workforce, and in regards to that, we have been increasing our graduate student throughput. Our population has grown 6.6% the last several years. That's even in light of the pandemic. And we've increased our graduate student throughput by 11.1% since 2013. So we are an engine for developing trained individuals that end up in your companies and institutions. So we're really excited to discuss partnerships today that involve these students, and you can maybe hire some of these folks in the future. So today I'm finishing my comments, but Deborah will walk you through. She's a pro at this, at a process to hear about what we're up to find out what your folks' needs are, and to make matches. And I'll stop there and thank you and have a great day.
Thank you very much, Dr. Lommel. That was great. Really appreciate your support. Next up, we have Dr. Carbone, who is the director of CIFER and the PSMCC. So go ahead and share your screen and you can bring us up to date.
Okay, can you hear me? Good morning, everyone. And really happy that you can all join us today. I have some exciting news for you. Today, CIFER is launching the Plant Soil Microbial Community Collaborative, or the PSMCC, to continue to strengthen our interactions with industry. I want to provide you with a brief overview of CIFER and what motivated us to launch the PSMCC. So CIFER is an interdisciplinary team of 17 scientists, shown here, spanning 4 colleges. Our vision is to provide fungal and microbial bioscience solutions for sustainable agriculture, health management, and environmental stewardship. Our faculty have very broad expertise, from our core understanding of fungal dynamics to chemical and environmental engineering, mathematical modeling, microbiology, soil science, evolutionary biology, ecology, and beyond. So the research talks this morning will give you a sample of the breadth and the depth of our work in CIFER. Now, the interdisciplinary interactions in CIFER, as shown in this pie chart, really emphasize the need for a cross-disciplinary approach to the agricultural microbiome. CIFER faculty are really committed to developing an integrated understanding of both the biotic and abiotic interactions that impact plant health and yield in agroecosystems. Some interactions, however, while not impacting yield directly, are important for food health and safety. And I want to just highlight one of these. Some chemical metabolites produced by fungi have a direct impact on animal and human health. So we want to study the entire gamut of interactions. Now, I want to make it very clear that an important outcome for us is the translation of basic research to application in the field. We strive for this. And this is actually what has motivated us to launch the PSMCC. So the PSMCC is an industry member supported program within CIFER with the mission to facilitate collaborative research between faculty and industry. We see many opportunities for innovations in modeling, predicting, and managing how microbes and plants respond to environmental perturbations. And what is unique about us is that we focus from the small-scale subcellular level to communities and populations to entire ecosystems. So we really want to understand the entire process, and we really need to do this to get a handle on what's happening. We see many opportunities for collaboration, and CIFER plans to host more networking events like this one to promote faculty-industry interactions. Hopefully, next one will be in person so we could really gel and talk more. But I want to point out some of our resources that would be of interest to industry, including strain collections sourced from North Carolina research stations. Steve mentioned this, and subsequently testing those strains for efficacy in field trials, for example, for identifying new candidate biocontrol strains. But we also have tools for big data microbiome analysis, which you'll be learning more about this morning. So this is all exciting, but how does the PSMCC work exactly? Okay, so there are essentially 2 models of sponsored research collaboration with university through the PSMCC. So first on the left here is sponsored research, whereby 1 or 2 companies directly fund a research program and share access to the results and any IP that's generated. We also have a second model, which is a consortium agreement. And here's where 3 or more companies co-fund a single project. The benefit of this model includes leveraging research funding and a reduced overhead rate. So as you can see from both of these models, our PSMC partnerships are completely flexible and negotiable. We really want to work with you, and we'll do it one-on-one in groups, whatever works. So I just want to end this saying we're really welcome the opportunity to start these collaborative projects. Mary and Deborah are available to answer your questions regarding the PSMCC. But I now want to pass it over to Dinah to describe yet another way that you can interact with us and the PSMCC. So Dinah, can you chime in here now?
Yes, thank you. Good morning, everybody. It's great to see so many faces this morning. I recognize several names, and like Deborah, there's a few I don't, so I will be looking you up. So my name is Dinah Schuster. I am the Senior Director of Development for the Plant Sciences Initiative. The building behind me is what we're currently working on building. But it's more than a building. It's also a lot of programmatic opportunities. And so as I look at some of the industry leaders that are on this call today, we're already in conversation with some of you about that. So as you can see, this is a collaborative opportunity. So the PMCC and the CIFR, and you can see we love an acronym, are great opportunities where we see lots of synergies where our faculty can be collaborative, and we can not only working within the college, we can also reach out across campus and into industry for lots of partnerships. So as Deborah is working with you all to create those partnership opportunities, I'm working with you all on your philanthropic interests. And so you'll often see our 2 names side by side because we would love to have your support. We think there's a way that we can work with you, and you're either going to work with her or with me or somebody on my team. So again, this is going to be a great morning. And I know that Mariana and Ignacio have put together great presentations for you all. And we just thank you so much for choosing this morning to be with us. And again, hope we can find ways that we can all partner together and do some really amazing things. Thanks so much.
Thank you so much, Dinah and Ignacio. That was very informative and very supportive. Thank you. And hopefully we'll get a lot of good collaborations and interactions going on. So I think we can dive into the research talks now. Our first speaker is Dr. Carbone again. You've already met him. He'll talk a little bit more about his own work. Go ahead if you're ready. The floor is yours.
Okay, I'm back. So I want to give you a quick overview of the biodiversity informatics pipeline that we're developing at NC State. And this pipeline has been in development for almost a decade now, with input from both academia and industry. So this platform really helps us overcome some of the major issues that we encounter in biodiversity studies. And here I'm going to list some of the challenges that we're facing. This is by no means a comprehensive list, but just to give you an idea why we need this tool. You may be aware that less than 1% of the microbial diversity is described, and this is true for fungi and bacteria. That means many sequences have no close match in databases. Molecular barcodes that we use, both in fungi and bacteria, it's better in bacteria, but in fungi, they frequently lack species level resolution. Species delimitation, we know, needs multi-locus data, particularly when we're dealing with fungi. We have lots of multi-locus reference trees that are underutilized. There's no mechanism to really use these in species descriptions. We need resolution to species for ecology. That's the unit of ecology. And actually for identifying new biocontrol candidates, we need to drill further down in the species. We need to look at lineages and individuals and populations. Classification is a continual state of flux, and I dare to say it's a mess in fungi, with names changing and disagreements as to what is a species. So this is something that we need to grapple with. Then there's phylogenies, alignments, and metadata. Try to combine all these and you'll have a hard time with hyphens and underscores, and typically it's a headache to do, and very few tools do this. Tree databases like TreeBase often don't include metadata, and you're often, when you're looking for this specimen metadata, it's found deep in a paper or as a PDF or supplemental material. And again, much of this metadata is not matching the names in the other data sets. And there's no data standards for phylogenies, alignments, and metadata, which makes it impossible to share these components of data as a unit. And we really need to do that if we're going to build on this. And then there's a need for microbial dark matter. This is all the undescribed, uncategorized sequence data that we typically just throw out. We need to fill the gaps in the trees where the known taxa are with this microbial dark matter, and we're not doing a good job of that. So this all motivated us to develop the TBAS platform. So what I want to do now is just walk you through this. First, what is TBAS? So TBAS is literally a tree-based alignment selector toolkit. Okay, it's a robust, multi-locus, the keyword, and metadata enhanced framework. And it's one of many tools in our Decipher, and I want to emphasize that, this is the Decipher platform developed in the center that we offer publicly and we license also. And this platform is really useful for tracking sequences, microbial dark matter, and associated metadata. So without showing you the tool, I wanted to give you a feel for how the user interacts with the tool. There's a data upload in a section that has to be done when you first run the tool. And here you would upload your single loci, your multiple genome FASTA files. These could be metagenomes. These could be short or long sequences. At the short end, you have metabarcoding. At the long end, for example, SARS-CoV-2 sequences. And it can handle DNA, RNA, or protein FASTA files. So the entire gamut of molecular data. But we also require the user to upload specimen metadata. This is key because without the specimen metadata, the sequence data has little sense. And so we want also users to upload counts data for metabarcode sequences. And I want to emphasize that this tool works across all domains: eukarya, bacteria, archaea, and viruses. We have reference trees that resolve at the domain level, reference trees for viruses, so it's completely expandable and flexible. So once you upload these input files, we're then ready to place these sequences on, and I emphasize, curated multi-locus trees. This is important because the trees have been linked to specimen metadata and alignments and are ready for placement. We have the option of clustering sequences by locus. For example, if you take the spike protein and coronavirus, we could cluster haplotypes in that region. We need to perform multiple sequence alignment because the uploaded files are unaligned, and we have MAFD. And then we have well-vetted phylogenetic placement tools, maximum likelihood, bootstrap, backbone constraint methods, or also de novo reconstruction of trees. Everything that you need for phylogenetic reconstruction of these unknown sequences. But we also provide taxonomic and functional guild assignments along with the phylogenetic placements. This is useful to have an idea of what these sequences are doing in the broader ecological community. So once the tree inference is done, we can then display taxon associations with different metadata categories on the tree. For example, we can link clades and lineages with specific biotic attributes. For example, here, host use, abiotic, or other metadata. But perhaps one of the most powerful features is that we could simultaneously view attributes of the unknown sequences that are uploaded with the reference taxa. So that's where the power of the metadata comes really into play. If you have the metadata for the reference taxa, you could then see how that compares with sequences that are clustering closely with those reference taxa. The other thing that is a recent advancement in our tool, the user can easily pan and zoom the tree with up to 36,000 tips. This makes it very easily to explore the tree and zoom in onto specific clades. The output, since we're reconstructing these phylogenies and the placement of these unknown sequences, we can readily generate distance matrices. And this is as shown here, and this is literally the pairwise distance from all the tips in the tree to each other. We could then perform principal coordinate analysis with these metadata categories. A number of statistical tests can be performed, and this is using just common tools like CHIME or PhyloSeq that are available. One of the one of the most useful features of this tool that we use since it was developed as part of the NSF Dimensions of Biodiversity is that we could test species boundaries hypotheses. We can zoom in onto specific clades, and using both single or most commonly multi-locus data, we can get a better feel for actually the breadth of a clade in terms of a species and where to start understanding where one species ends and one begins. So this is a powerful characteristic of this tool. So after all this, the output is compressed into a metadata enhanced phylo-XML or MET file. This cross-links the phylogeny, the alignments, and the specimen metadata here in a single file. This is important if you want to continue to build on these alignments and the metadata. This is our standardization of the data that really makes this tool powerful. We also provide detailed reports of phylogeny-based assignments, and we output all the distance matrices, MEP, MUIC files, biome files, so that users could actually integrate this tool in their existing pipelines. We actually have REST features that you could send commands to TBAS, retrieve phylogenetic placement information, and continue with your pipelines. So this is an overall tool that really is empowering us to understand microbial diversity across all domains. Now, I described the tool it's freely available, it's open to the public, and you can access it, it runs on the high-performance supercomputing machines in the US, known as Exceed, but users also have the option to purchase a license. We Dockerized our tools, which means that you can use the tool within your own firewall without connecting to the internet, and so all your data is contained within your own walls. And we have both single and multiple user licenses. Since licensing version 2.2 of TBAS last year, we've had one company, Mako Medical, license it for analyzing SARS-CoV-2 sequences. And if you're interested, I'd be more than happy to show you what TBAS can do for your data and your metadata. So with that, I'll just stop and answer any questions you might have.
Thank you very much, Dr. Carbone. Deborah, are there any questions for Ignacio?
There's a general question, which is, will the slides be shared? And the answer to that is yes. And other than that, there are no questions in the chat.
All right. Thank you. Next up, we have Dr. Oliver Bars. So go ahead and share your screen and jump right in.
Okay, do you see my presenting mode?
Yes.
Perfect. Thank you so much for the opportunity to talk here and present some of my thoughts and my program here, which is starting. I've been here for now about 3 years in the Center for Integrated Fungal Research and also in the Department of Entomology and Plant Pathology. So I'm talking about trace metals and how they can drive rhizosphere microbiome interactions and exudation of chemicals. One of the main problems, what I gather in agricultural biologicals, is that they often perform differently and unpredictably in various soils and with various plant hosts. And I think that iron can be a critical driver of this, iron and phosphate. And they provide a good handle, in my opinion. So iron and phosphate have one thing in common, is that they are abundantly present in soils, but they present a problem for biological acquisition because they are chemically bound to soil particles and not really accessible readily. So plants and microbes use their biochemistry to acquire sufficient iron and phosphate by secreting chemicals. And, you know, basically the idea is that it's all pH dependent. At a high pH, iron precipitates as iron oxides, and you know, you can see this when you make an iron solution basic in a test tube, and over time this can age into minerals. And these are very hard to get for biology. And the idea is that biology uses various metabolites in order to solubilize the iron and reverse this reaction and take up iron. At a very high pH, iron is extremely insoluble and extremely hard to access. And so you need a pH of roughly about 4 or 5 to get enough iron for plants. The other thing is phosphate. As you may know, is also not readily available, and particularly at low pH, it can either be bound to iron and therefore not be available, or at high pH be bound to calcium. So the ideal pH for phosphorus acquisition is between 6 and 7. And there is no pH where phosphate and iron are readily available. And you can see here a nice illustration of rice plants grown in a calcareous soil, and this is the control. And then here is added a phytosterophore, which solubilizes some of that iron via secretion of this phytosterophore and helps the plant grow. So iron and phosphate are both strong elicitors of exudation of metabolites, and these are these can be organic acids in the case of phosphate. And you may know this maybe a lot of people screen for beneficial bacteria using agar plates and look for example, phosphate solubilization. And you see this as a clearing zone where the bacteria make organic acids and it clears the phosphate precipitate around the bacteria. Iron acquisition, in contrast, whereas phosphate acquisition uses basically a couple of organic acids, iron acquisition is much more specific and diverse because chelators can be specifically targeted towards a cation, whereas it is more difficult to target phosphate. So iron acquisition is very specific, and there's hundreds of structures that are targeted towards iron acquisition. And there's even 2 different chemistries. Either you chelate iron 3 for uptake, which is done by bacteria in plants, or another strategy is to reduce iron 3 to iron 2, which is more soluble. So these can also involve organic molecules that are secreted. Now, why does this matter? So there's all these specific structures it is unclear. So this is a big question. While, there's a lot known about how important iron is for plant microbe interactions and immunity and microbial competition, what is really unknown is how this diversity of structures plays into this. All of the studies are usually done with one structure that's commercially available, desferrioxamine B, or in like individual, test tubes, but not much is known at all about how different the diversity of structures affects things. So it is known that there's a tug of war for iron between bacteria. Bacteria can also compete with plants or help plants to acquire iron. Some of these siderophores can be specific signals to the plant so that the plant understands there's a certain bacterium. Exudates can also be signals to microbes and act as antimicrobial agents, but how this diversity affects all of these specific reactions is really not known or poorly known. So what I do in my lab is, and this could be then used, if we understand this, I think this could be critical to develop effective agricultural biologicals. The synthesis of these compounds is usually genetically tractable, and microbes that make these can be more competitive, help plants get more iron, or help plants help to trigger an immune response from plants. So soil chemistry, interactions, and iron are closely linked. So what I do in my lab is I first want to identify the structures produced in response to iron limitation and then understand their interactions with microbes. Here is just one example where I started with plants releasing exudates in response to iron limitation. I wanted to know specifically for tomato what they are producing. And you can see here so-called charged aerosol detector trace of tomato exudates in different conditions, replete in blue, phosphate limited in purple, and then iron limited in black. And most of the exudates, and I see this with bacteria and plants, are produced under iron limitation if you look at secondary metabolites. and that is so iron is a very strong elicitor of exudation. We identified these exudates from tomato, which are primarily coumarins that have been previously described in Arabidopsis, and then we assayed them with bacteria wilt, which is a problematic pathogen for tomato. It has a strong effect on its biofilm formation, as you can see here with added compound versus another microbe. Doesn't react that way, so it has a selective effect. And we also see this then if we do, for example, growth assays where this bacterial wilt is completely inhibited by this compound, versus another microbe may grow somewhat or better. So there's a selective effect of these exudates that are iron induced under iron limitation on the microbial community. On the other side, there's bacterial exudates, and I use my own software and bioinformatics to find these compounds, these siderophores. This has been published and is a very effective tool to detect iron chelators in solutions and in extracts. And I've started looking at now a 7-member synthetic community with maize. Synthetic communities that are simplified have the big advantage that I can track metabolites. Metabolomics always has the problem, you don't know who is producing a certain metabolite. And with a smaller community of 7 microbes, I can understand who produces the microbe, and we can really, for the first time, look into the siderophore interactions in a community. And I have started to identify the siderophore produced by some of the members of this community, which has been published in PNAS 3 or 4 years ago. The next step would then be to understand the interactions and competition, signaling, and so on in this community. Finally, I measure also real soil samples and have developed methods for measuring some of these iron chelators in soil samples, and we found them in North Carolina samples, even in soils that are relatively acidic, further suggesting the importance of siderophores across a wide range of soils. And so I think iron is a very ubiquitous driver of plant microbe interactions, not only in calcareous soils, but even in soils here in North Carolina. And that's my short presentation. I'll take any questions.
All right. Thank you so much, Oliver. Well done. I think we don't have time for questions right now for Oliver, but he will be in a breakout room at the end of the sessions. So next up, we have Dr. Wei Shi. Wei, if you're ready, you can feel free to share your screen and begin.
Wei, we can't hear you. Are you still muted? Wei,
Okay,
I'm
sorry.
There we are. Very good.
So I'm soil microbiologist in the Department of Crop and Soil Sciences. My presentation is about turfgrass root-associated microorganisms. As we know, root-associated microorganisms has been considered as the extended plant genome because microbes play important roles in plant adaptations to the environment, such as nutrient acquisition, stress tolerance, and the defense against the pathogens. Therefore, using microbes to improve plant environmental fitness is promising for reducing the heavy reliance of managed systems on synthetic chemicals and limited resources, such as water. This is particularly important for turfgrass which is an intensively managed system with fertilizers, pesticides, herbicides, and irrigation for maintaining a healthy landscape. One of the strategies to develop microbe-based technology in agriculture is to screen dominant microbial colonizers, understand their beneficial effects, and then formulate a recipe of effective colonizers. So I'm going to share some early research progress of this strategy applied in turf in my group. So the question that we were asking were what microbes dominate turfgrass root, endosphere, and the rhizosphere. Did the microbial dominance vary with turfgrass species, turfgrass sites, management practices, and growing seasons? To answer these questions, we collected dozens of intact turfgrass root and soil cores from different turfgrass species, turfgrass sites, and management practices, and also different growing seasons. Then this intact cores was separated into respective bog soil, rhizosphere, and grassroot samples. After DNA extraction, microgene library preparation, and high-throughput sequencing, we use bioinformatic tools to parse microbial community compositions. This is a principal coordinate analysis of bacteria and fungal communities. Different colors represent data from endosphere, rhizosphere, and bog soils, respectively. Different symbols represent data from different turfgrass species or cultivars, management practices, turfgrass sites, and growing seasons. So we can see clearly that endosphere bacteria and the fungal communities were well separated from those in rhizosphere and the bog soils. And this compartment-based separation was a little affected by grass species or cultivars, turfgrass sites, management practices, and the growing seasons. Here I use a Venn diagram to show overlapping rates of dominant microbes among the three compartments. We can see that overlapping rates were lower for both bacteria and the fungal communities, indicating that dominant bacteria and the fungi differ largely between bog soil, endosphere, and the rhizosphere. For example, at the genus level, Pseudomonas was dominant in turfgrass root, endosphere, but Gymnobacterium, Pseudomonas, and Pseudogymnoscus were dominant in turfgrass rhizosphere. We also examined environmental factors and their impacts on dominant microbes in bog soil, rhizosphere, and endosphere. As predicted, dominant microbes in bog soils were highly dependent on environmental factors such as soil properties, turfgrass species, management practices, turfgrass sites, and sampling times. But dominant species in rhizosphere and endosphere were later affected by ecological factors, but the presence of turfgrass. So in summary, a few microbes dominate in turfgrass root, rhizosphere, and endosphere. And these dominant microbes show greater compatibility to a variety of turfgrass species. Based on genome prediction, these microbial colonizers are potentially involved in turfgrass nutrient acquisition and the environmental fairness. So we are going to further evaluate their responses to environmental factors in future. So that is what I have. Any questions?
Thank you so much, Wei. Deborah, do we have any questions for Wei?
There are no questions yet,
Okay,
but don't forget, Wei will be in breakout room number 5
that's right.
post-event.
All right, well, I guess we'll move on then. Next, we have Dr. Peter Ojambo. So Peter, go ahead and share your screen and introduce yourself. And just make sure you're unmuted.
All right, can you hear me? And can you see my screen as well?
Yes.
Okay, thank you. So, good morning, everyone. So, today I'm just going to be talking to you guys a little bit about some of my work that I've done on Kikabi downy mildew and how that work has actually resulted into some partnership with the industry. This has been work that has been conducted over so many years by my graduate students and some collaboration with the members of CIFER as well. Okay, so how do I advance this? All right, so Kikabi downy mildew, for those of you that don't know, the disease is caused by an oomycete called Pseudotheranos pracubensis. The symptoms are very varied depending on what Kikabi you are actually dealing with, and the disease has a wide distribution and pretty much is present where Kikabis are found, and warm and humid weather actually does promote disease development. But as far as control is concerned, we have very limited control measures. We don't have any resistant cultivars, and there's a lot of reliance now on the use of fungicides. And the disease forecasting has been developed primarily just to help growers determine the initial spray. And so if you look at the status of the CDM in the United States, it's primarily in the eastern part of the country, and that's primarily because that's where environmental condition favors disease development. And then there was a resurgence on cucumber primarily in 2004. Since 2004, the disease is now occurring every year, and it can cause devastating losses in absence of disease control. And in California, since 2007, it occurs annually every year as well. And so there is resurgence. The main question was, okay, what caused this resurgence? And so we decided to develop some projects here to be able to figure out exactly what happened so that we can actually help our growers better control the disease and figure out exactly what they need to do. And so what we did, we went on a series of adventure, collecting isolates, throughout the country from different cucurbits, and then we went ahead and, characterized them based on the virulence and mating type. We conducted a lot of work on comparative genomic analysis, and then what we found out was that there were new pathotypes that were actually present that were not here before. And so we knew there was also another new mating type that was present here, and the new lineage was also present here as well. And so based on that, we were able to get a lot of information that came into place, because we now had a collection of isolate that had been characterized. We have genotyped these, we have sequenced them, and we had information about, how virulent they are, how aggressive they are, and we also were able to generate information about how, resistant they are to some specific fungicides. So this biological resource, we found it as very, very useful in so many ways. And this opened up new opportunities for us to collaborate with the industry in this regard. So our first set was just to work with the Bayer Crop Science. They wanted to figure out exactly what was happening to some of their products. And this work was done to look at fungicide resistance with Bayer and the Valent. And the goal here was to link fungicide resistant pathogen biology, because if you could link, based on the genomic resources that we have, could link that to a specific phenotype of interest, then of course, it opens up new opportunities to better manage the chemistries, design them in such a way that it can target specific strains of interest out there in the field. So we did that, and then we went ongoing now with subsequently, we developed another project that's actually going on right now with Bayer Crop Science and Valent. And in this particular case, we are looking at the biology of the pathogen. We're looking at the genotypes, and then we're trying to develop mutants and try to predict, the resistance that can actually evolve over time with some of the new chemistry they're actually developing. And then we have a similar project now currently also with the ag biome and trying to evaluate efficacy. And this is very targeted for both companies. It's very targeted, without giving a lot of details, that we know the kind of information we have about the isolates to specific resistant to specific fungicides. And so we are looking at those ones that are actually have cross resistance, they have specific lineages or mating type, and then we're screening them to their biological. So this is more targeted so that the information that's actually generated is actually very effective in terms of them designing new fungicide chemistries as well. And so this would not have been possible, because of without the resource that we have now here at CIFAR. And this took us a lot of time and effort to actually generate, but it has opened up new opportunities, that are ongoing now. That that we're now partnering with the industry, so that they can actually take advantage of some of the things that we've been working on for the last 10 years. That's now well curated, well characterized, so that whenever you, for example, you want to look at new chemistry, for example, you can tell us what you want, whether it's a specific mating type or lineage, or resistant to a particular fungicide or a particular pathotype, and then we can actually curate that for you and then test them so that you can get more informed information about the product they're actually looking at. For that, I'll take any more questions. Any questions that you might have. Thank you.
Thanks, Peter. That was great. Do we have any questions?
There's nothing in the chat right now, but Peter, I wonder if you'd talk I love this last slide, of course. Can you talk a little bit about the value to your program to having these industry partnerships?
Well, quite a lot. I can say it has been very valuable for me. For example, with Bayer on board and Valent, and ag biome, we have been able to this ongoing project. We have been able to establish partnership, in terms of training, and also we are utilizing some of their bioinformatic groups. So they are bringing also information to us that we didn't have before to better improve the things that we actually are developing. And this partnership, for example, has enabled us to exchange information back and forth. And that has been enriching us in CIFAR, and as well as in Bayer, as well as in Valent. So this partnership, I find it goes both ways, and we have been publishing papers as a result of this. So it has been something that has been made possible because of the effort that we put in the beginning, the last 10 years. Now that we have this collection, that we can now, is a resource that people can actually use and utilize to be able to answer specific questions of interest to them. Yeah.
Thanks, Peter. There's nothing else in the chat right now.
All right, then let's move on. We're actually a little bit ahead of schedule. So Dr. Nathan Crook is up next. If you're ready to share your screen.
All right, everything visible and you can hear me okay?
Yes.
All right, thank you very much. So today what I'll talk to you all about are our efforts to develop genetic engineering tools for plant microbes, and specifically those that are colonizing this maize community, which Oliver so kindly introduced before me. And so one of the key challenges that often happens when you develop a bioinoculant is that a microbe that you may find looks great in the lab, but when you try to apply it to a real plant with environmental microbes colonizing it, the transfer doesn't work. This strain is outcompeted by native microbes. And so this could be due to competition, metabolic interactions, incompatibilities with the nutrient environment that is there, many potential factors. And so what this has led to, and this is motivated by discussions I've had with Katie Barnhill-Dilling at the Genetic Engineering Society Center at NC State, is that a lot of growers consider bioinoculants to be some sort of snake oil. They don't really work. And so we need to overcome that perception. So in order to overcome that perception, we need actual mechanistic understanding of how these interactions work. And that is done by model systems, gene level understanding, and also in order to get that gene level understanding, we need to perform genetics on these bacteria. And so this is also motivated by interviews I've had with industry, that they find that they can't even transform the microbes that they want to work with. And so we're going to overcome those here. And so the system we've been working with has been this maize synthetic community. Oliver so kindly introduced it. This is sort of how it works. You have two glass cylinders that you tape together and you plant a seed in there. And this is just community dynamics of those 7 member communities over time. The cell mix is roughly equal, but then they have their dynamics over the course of 2 weeks while that strain germinates. And this is a nice community because it also has the ability to resist fungal colonization. And so it has some interesting biology going on there. So we've had 2 focuses in our lab related to this community, the first of which are genetic tools for these strains. And so what we're trying to do in our work is to develop plasmids that are able to more efficiently mobilize into these strains than the plasmids you might get off the shelf. Another thing we're trying to do is develop a promoter library. So how we tune gene expression in these organisms to levels that you would like. And this is useful because what we can do with that promoter library is develop gene editing tools. So if you want to investigate the function of a gene, you need Cas expression to be well tuned. And in order for that, you need a promoter. So as preliminary data, what we've figured out is that these different microbes have vastly different transformation efficiencies, and that efficiency depends upon where you get that DNA. So if you get that DNA from E. coli, which is what we often do, that transformation efficiency can be relatively high, 10 to the 6 per microgram, or 0, depending on the strain. However, once you transform that DNA into your organism, that organism methylates that DNA to give it a relatively high transformation efficiency. And so what we're trying to do in our work is develop plasmids that simulate these methylation patterns and allow us to get higher efficiencies. Our second focus has been understanding colonization in these microbes. And so the way we propose to do that here is to perform in planta selections, and then followed by genome sequencing and annotation of the mutations that we get. This is motivated by prior work that I've done in the mammalian gut, where probiotic microbes that don't colonize our guts, actually do evolve during their passage to improve their fitness. And so we think that plant-associated microbes are going to be doing the same thing. We can understand that process in 2 different nutrient conditions, iron replete and iron limited. And so we're going to sequence them and identify what those mutations do. So this is a timeline, and the deliverables here are broad. So we can do this for our maize synthetic community, or if you have microbes that you would like to study further, we can do that too. The student who's working on this is John Van Schaik. So again, he's being trained by the PSMCC, and we're all working with Dr. Manuel Kleiner for the plant experiments. So thanks, and I'll take any questions you have.
Thanks, Nathan. That was great. Deborah, are there any questions for Nathan?
We do. We have a really interesting question. Nathan, the question is, what is your opinion of the commercial viability of GMO microbes as an agricultural product from the perspective of US and ex-US regulatory bodies?
Yeah, that is a very good question.
That's the one, right?
Yeah. So what we've been looking at are the regulations in the US. We haven't looked too much at ex-US, but there is a regulatory framework for those GMO microbes, and I think it is primarily EPA that regulates them. And the notion there is to you perform an assessment about whether that is a harmful change. And I think there will also be some biocontainment features that you have to look at as well. There is a path, the notion is there's a path forward. And there's also a company that we've been talking to called Join Bio, who is developing GMO microbes. So they obviously think that this is a viable commercial strategy. But the important note here is that gene editing is not just to develop a gene edited product, it's also to understand a natural microbe that you might be more interested in. If you find that there's a strain or genetic change that you like, you can then look for that type of gene in natural microbes to avoid that designation.
Thank you. And that question was actually doubled down. So
okay, good.
But great answer. Thanks, Nathan. That is all that we have in the Q&A box right now.
Okay. Well, that's the end of our first set of research talks. So I think we can take a short break and we can come back here at 10 minutes after 10. And we will then start off with Dr. Buckler, who's up next after the short break. Feel free to please stick around and talk to our speakers if you wish. And the breakout rooms will open after the second session. All right. So thanks and see you again shortly.
Let's see if this gets going. So can you see my screen?
Yes.
All right, fantastic. Well, so thank you very much for giving me the opportunity to sort of present what we're working on and where I think there could be some interesting opportunities thinking about soils and crops. So our lab works with fungi. And one of the sort of the main directions in the lab now is we work with sort of what I would call zoosporic or early diverging fungi, such as Spizellomyces. So you're seeing a nice time-lapse movie here. And yes, it is a fungus, it's crawling, and it actually can swim, right? And this is a feature of these zoosporic fungi, and hence their name. And so you may say, well, what are these zoosporic fungi and why do you work with them? And I just want to sort of orient you a little bit here with a sort of fungal tree of life. And what you'll notice is, of course, that most of the fungi many of us work with and that play roles in the soil are here, sort of the higher fungi, but there's early diverging lineages of fungi. And these are loosely what I'm going to call chytrids. And these blue ones all have these zoospores. So they form spores that can swim and crawl. And what I want to point out, and the reason we're studying these, is that these have ancestral features that are found in animals and a lot of protists, all right? So they're fungi that have not lost the ability to crawl and that have not lost the ability to swim. Yet, as I'm showing you up here, they have a typical life cycle that also has fungal components. For instance, they'll germinate, they'll have fungal cell walls, they'll grow sort of hyphal-like structures into organic matter or also as pathogens into living matter. And so, not surprisingly, these still have these ancestral features because you often find these still in aquatic environments, life evolved in the oceans, and you also find them in sort of soil biomes. And so of relevance to the community today, some of these are parasites or disease vectors. So you may know or have heard of Opidium, which is sort of a disease vector for cucurbits, or Syncytium, which is a pathogen of potatoes. It's also a pathogen of animals like Batrachochytrium dendropatitis of amphibians. So the reason we study it, and in particular what I'm going to show you, is we've been working with one model organism called Spizellomyces punctatus. And the reason we work with it and people are interested in chytrids is that they have a lot of not just ancestral features, but they also have a lot of ancestral genes that you find in animals or the animal ancestor that have not been lost yet. So most sort of higher fungi have lost these. And so we are interested in working with them. And what I'm presenting you today is sort of a novelty in our lab is in order to work with this, we want to be able to manipulate the genome of a chytrid, which hasn't been done in any other chytrids. And so what I'm going to show you in the next couple of slides and open directions that I think are exciting for soil biologists are we got agrobacterium to work to transform Spizellomyces. This is recently published work. We've identified drug resistance markers. We've got fluorescent proteins and promoters that work really well. And we can actually transform our chytrid. And here I'm showing you a nice movie where we've actually labeled LifeAct, which binds filamentous actin with a fluorescent protein TD tomato. And you can actually see these crawling zoospores who are sandwiched between a cover slip and an agarose pad. You can see that it's the filamentous actin is all localizing to the leading edge, as you might have expected for a crawling sort of amoeboid-like state. So this is exciting for our lab, but I think in particular, sort of the directions that I'm interested in moving forward is there are a lot of sort of opportunities here in which we've got promoters, we've got the tools with which we can transform chytrids. And so there's some important commercial ones. So for instance, thinking of alga culture for biofuels or other things, they have a big problem in these open pools is that chytrids are algal parasites, and they can come in and wipe out entire populations and really affect yield and problems. Another one is Syncytium, a chytrid that is causes disease in, say, potatoes, sort of potato warts. And a direction that we're interested in, because we're in the vet school here that we've started moving in, is there also turns out that in the rumen microbiome, there are very few eukaryotes, but there's one that's strongly present, and these are anaerobic chytrids. And so we have ongoing work right now where Andrea Vu in my lab, a new postdoc, she's actually worked in oomycetes. And so she's taking her expertise from sort of zoosporic non-fungi, and she's been able to get electroporation to work rather than agrobacterium transformation in Spizellomyces. And we're very interested in moving forward and studying and transforming the anaerobic chytrids that are in the rumen microbiome. And the idea here is to study them, but also potentially to manipulate them for probiotics and improving sort of nutritional yield in ruminants. So with that, I'll gladly take any of your questions. Thank you.
Thank you so much, Nicholas. Are there any questions?
Nothing right now in the chat. And don't forget, Nicholas will be in breakout room number 5 after the event.
All right then. Okay, we can move on then. So Johnny Balidion is up next, and he is a PhD graduate student. Yes, he is a PhD student in Dr. Mark Cubetta's lab. Johnny, if you're there, go ahead and share your screen.
Okay, thank you, Dr. Carbone. Can you see my screen?
Yes.
Okay, good morning, everyone. Thank you for giving me a chance to share my research this morning. So basically, I'm a PhD student from the Entomology and Plant Pathology Department working with Dr. Mark Cubetta, and I'm sharing to you today about my project on looking at plant water availability as a driver for endophytic fungal activity in a system that I am working on from tropics. So ever since I started my research career, I have always been interested with the interactions of biotic and abiotic stresses in plant diseases. So I started with bacterial leaf blight in rice and its interaction on temperature genes, resistance genes, basically. Then I looked at interaction of synchroviruses with R genes, followed with disease severity of 2 interacting viruses on orchids, Philippine native orchids and cultivated orchids. And recently, we're looking at a system where there are 2 interacting pathogens, Ceratobasidium theobromei. This is a rhizoctonia-related fungi, that's why I'm working with Mark, and Lactobacillus theobromei and its association with a dieback disease in cacao in the Philippines and Southeast Asia, which is endemic. So basically, we are interested at looking at the effect of water stress on latent pathogens such as Lactobacillus theobromei, and we think that water availability or water stress would have an influence on symptom severity and eventually disease development. So we think we are looking at comparing the symptoms and fungal colonization rather in seedlings exposed to water stress. So basically, I'm looking at well-watered and water stress conditions with inoculation of this latent fungi Lactobacillus theobromei, and we think that water stress will enhance fungal colonization and symptom expression in seedlings which are subjected to water stress than those which are not subjected to water stress or the well-watered conditions. So basically, we would like to look at this system in cacao, where we introduce our fungi previous to the introduction of the water stress and incubated for a series of, let's say, 3 to 4 weeks, and eventually look at fungal colonization and disease severity and see if water stress would increase fungal colonization and disease severity in this cacao genotypes. So we have tried looking at this system also in animal crops with a similar endophyte. And now we are looking at the, cacao as another model system for this latent pathogen in woody crops. So another study that we would like to look at is actually the biological based management of this latent pathogen in cacao or other woody crops. So we are looking at the effect of beneficial endophytes on Lactobacillus theobromei cacao interaction, where we are going to introduce the beneficial endophyte first prior to introducing our latent pathogen. And we think that beneficial endophyte inoculation will reduce fungal colonization of Lactobacillus theobromei and disease intensity in cacao seedlings compared to the non-inoculated plants with a beneficial endophyte. So basically, this is a summary of the system that I would like to look at too. So I would be introducing the beneficial endophyte while growing the plants prior to inoculating our latent pathogen, and see if the fungal endophyte inoculation would influence the movement of this pathogen despite the presence of the water stress, and eventually look at microscopic examination of the colonization of the endophyte in the presence or in the absence of this latent pathogen. So basically, this would be the setup. And we are looking at several endophytes to be used in this study. And all of these are conducted actually in biosafety level experiments, because these are all important organisms from tropical conditions. And I am very thankful that the NCSU have the excellent phytotron facilities and BSL-3 containment facilities. And I'm working with Carol and Joe in that facility to accomplish all of these experiments. So if you have any questions, I would be happy to take them now or later at the breakout rooms. Thank you.
Great. Thank you, Johnny. Deborah, are there any questions for Johnny?
Nothing in the chat yet.
Okay. Anybody jumping in? No? Okay. So let's move on. Next, we have Dr. Ben Callahan. Ben, if you're ready, go ahead. Is Ben there? Okay.
I'm here.
Oh, good.
Good to see everyone today. So today I'm going to talk about long read sequencing, or a specific flavor of it that I'm going to refer to as highly accurate long read sequencing, with a focus on kind of amplicon sequencing for assaying bacterial and fungal communities. So just as a very quick primer, so often the way that we measure microbial communities kind of in situ is with these sequencing-based technologies, where we take a whole community sample, we grind it up, turn it into DNA, sequence it, and then use those sequencing reads to sort of reconstruct a census of the community. So this is now a very widely used technique that's sort of synonymous with, microbiome science. So there are two ways to do that. So one, you can do shotgun sequencing, you just sequence everything. The other is that you target some particular marker gene that is informative and relevant to the taxonomic group you're most interested in. So this has benefits. You're able to target maybe sometimes a very small portion of the sample that you care about, like viruses within a human sample or something like that. It's also limited in that, here's an example. We have a genome, which is maybe many megabases, and we're amplifying up, a 300 base pair bit of DNA, because that's what fits on a sequencing read. So this is, very widely used. And one of the things that's important here to keep in mind is that accuracy is really critical, because when we're looking at complex communities, we don't know how many types are really there. This is not, you can't get too far with a noisy sequencing technology, because you start to get confused. Are these errors that I'm seeing, new biology, or just errors that are cropping up at 1 or 2 or 3%? So this has been dominated by short read Illumina sequencing, both for cost purposes and because Illumina sequencing is very accurate. More recently, these long read technologies have been developed that are really fundamentally different beasts. Their read lengths go out to hundreds of kilobases, but their per base error rates are just completely different. So where Illumina, nowadays 0.1% error rate is kind of the right number for Illumina. For PacBio and Oxford Nanopore, maybe more like 5 to 8% error rates, per base error rates, but just completely different sort of sequencing technology. Now, more recently, just in the past 3 or 4 years, what I will describe as a new sequencing technology has arisen, which is highly accurate long read sequencing. And there's a fundamental strategy here, which is we're going to build a consensus long read from multiple reads of a long DNA molecule. So, okay, any individual long read sequencing technology has high error rates, but if we can get multiple reads from the same DNA molecule, now we're in business. Now we can have long reads with very high accuracy. And there's currently sort of 2 well-supported commercial solutions for this that are out in the wild right now. So the first is PacBio circular consensus sequencing. This has been around the longest, and it's been rebranded in the past couple of years as hi-fi sequencing, if you've heard of that. And the second is this is a synthetic long read technology from this company Loop Genomics. And so there, basically they're using a bunch of short reads that are all tagged with a UMI saying which DNA molecule that it came from to create synthetic long reads from these short reads. And so they're not going to go out to hundreds of kilobases, but they'll go out to tens of kilobases. And just this number on the bottom is what I want you to notice. They have a lower error rate than Illumina does. And how much lower is sort of piqued our interest. And so we got involved in kind of evaluating and validating these technologies. And just to sort of show you again why these are effectively a new technology. So on this little plot, here's read length on the x-axis, accuracy on the y-axis. Here's the short reads we're used to. Here's the long reads we're used to. And these highly accurate long reads just are inhabiting a different space, you know, across these two important parameters for sequencing technologies. It's a different technology that is still, you know, people are learning about, and there are a lot of opportunities to use this type of sequencing technology in the future, I think. So to do an evaluation of this in a microbial community, we used this, what's called the Zymo mock community. So they took 8 bacterial strains, they mixed them together, and they will send it out to you in the mail. And so what we did was we did the most common sort of amplicon sequencing profiling in bacterial communities. We did 16S sequencing. But what we did full length 16S amplicon sequencing using the PacBio Hi-Fi, so highly accurate long reads. And these are sort of the results. So just to explain what these results are, every one of these bars is a specific ASV or allele of the 16S gene. They're all joined together with the strain from which they came, and the genus of each strain is in the top of these facets. And the relative abundance of that allele, scaled to the genomic abundance, is the y-axis. So in the Bacillus strain in this community, we saw 4 copies of this allele, 2 copies of the second allele, and 1 copy of each of these other alleles. And so what you should take away from this is we were able to perfectly recover all of the full-length 16S sequences in this mock community, and that without any false positives, and that includes every unique allele of the 16S gene, which is a multi-copy gene in most bacteria, every single allele of all of these strains as well, in their appropriate ratios. So if you look at both PacBio Hi-Fi and LoopSeq, so that's the technology from Loop Genomics, they have very high per base accuracy. So we measured in amplicon sequencing applications PacBio to have 99.96% per base accuracy. LoopSeq was even another order of magnitude, 99.995% per base accuracy. The errors are well behaved. You know, their quality scores are informative, and the errors are distributed pretty uniformly across the reads. Substitution errors, which are easier to deal with, are the dominant error mode. And this scales out to at least 6 kilobases, and the accuracy is maintained in complex samples. And so our evaluations of these two technologies are both published and are available in the literature. So where does this technology fit in? So thinking now about profiling microbial communities, there's sort of 2 axes that I'm thinking about. So how much non-target DNA is in my sample? And the other axis I'm thinking is how much resolution do I want to get when I'm doing my community profiling? Do I want to identify things down to the species level or lower? Or if I can just identify what family or phylum they're from, is that fine? And so highly accurate long read amplicon sequencing really fits in when you need higher resolution and when there is a significant amount of non-target DNA. That's where it sort of seems to have a good value proposition in this community profiling application, because you get the benefit of PCR targeting of the taxa you're interested in, and you get much higher resolution by having a lot more information. You know, I can be amplifying up an entire ribosomal operon that's 5 kilobases long, instead of trying to deal with a 200 nucleotide piece of the 16S gene. And just to very quickly touch on a couple sort of proto examples of this, you know, we applied this to some retail meat samples, and we're able to identify to the species level the foodborne pathogen species that the CDC has identified as of interest. And we can extract in these real samples, again, the complete allelic complement of the 16S gene. So these are all the alleles in the V2 region from C. perfringens. And really, we have the entire gene. We just only show the V2 region to make it easier to see. In the Zymo mock community, we were able to say more than just than just this was E. coli, we were able to say this was E. coli 0157H7. So that's an enterohemorrhagic major threat to foodborne safety. And that's a lot more informative than saying this is E. coli, most of which are fine. And in human fecal samples, we're able to distinguish distinct strains of E. coli, both between subjects, it's 2 different subjects on the top and bottom time courses, and within a subject. I did something to my slides there. There are probably a lot more applications for the sequencing technology outside community profiling. You know, high-ploidy genetics, like in plants, tumor sequencing, things like this. And really, I'm talking about technology developed elsewhere, so by these companies. So where we come in a little bit is that we developed the software Data 2 that supports the processing of amplicon sequencing. And so as part of these evaluation projects, we have been updating our software to handle and work with long read sequencing. So thank you, and just acknowledge other people that I worked with, and in particular, both PacBio and Loop Genomics were collaborators on the projects that I did, looking at their technologies. Thanks.
Great, thank you, Ben. Deborah, I think you said we have a question for Ben.
Yeah, we've actually got 2 at this point. First question is, what is the critical error rate that needs to be achieved to query a microbiome? When do amplicon errors overwhelm any accuracy gains for the base reading sequencing tech?
So I'm going to interpret this as, when does the errors made in the PCR process overwhelm the errors made by the sequencing instrument? And so these sequencing technologies are reaching that point where the most important errors are really coming from the PCR process often. There both are substitutions by mistakes made by the polymerase. There also can be the construction of what are called chimeras, where there's incomplete extension of a molecule in one round of PCR that then is extended with another molecule later. What's interesting is that LoopSeq and sort of these UMI methods actually have a way to almost completely get rid of chimera errors as well, by just making sure that the UMIs are consistent along the read that you create at the end. And so they help to guard against those kind of errors in a really powerful way. And so that's another advantage that I didn't talk about here. I was just focusing on per base errors. I hope that answered the question.
Thanks, Ben. There's another one, and I put these into your chat because I know they're easier to read sometimes. Have you used this technology to determine absolute abundance of 16S alleles in a community? For example, estimating the absolute abundance of a pathogen within a microbiome with a background of closely related strains.
So one of the things that these can do, as compared to short read amplicon sequencing, is they'll be able to distinguish between those closely related strains pretty effectively. I mean, there's always some limit, but depending on how closely related, they're more likely to distinguish them. Long reads don't solve the issue of absolute abundance. So in order to get absolute abundance measurements, you need to supplement your relative abundance measurements you get from either shotgun or amplicon sequencing with some measure, either a spike in that you measure absolute abundance relative to, or sometimes people do like qPCR of total 16S as a proxy for this. But that's really a challenge and a discussion that's external to whether you choose to use like long reads or short reads or highly accurate long reads. And I hopefully answered the question. I start forgetting what the question is by the end.
Right, and that's so you should see these, Ben, in your chat function, just so they'll be easier for you to read as well. One more for you, Ben. Do you have the technology in-house, and are you planning to provide it as a service?
So again, the core technology here is really developed by these companies. So I don't want to claim any credit for developing, PacBio Hi-Fi or Loop Genomics LoopSeq. What we really revolved in was doing kind of an independent evaluation and really trying to nail down in detail the error modes in those technologies. And we think we did a decent job of that. And then we also have the software that will help you work with that kind of sequencing. That's open source software that's available as an R package and through the Chime 2 platform. And so that's available to anybody who wants to use it. That part that we created.
Thanks, Ben. Mary, that's all we have in the chat.
All right, sounds good. Great, thank you. Next, we have Dr. Shuijin Hu. Shuijin, if you're ready, please go ahead.
Yeah, I'm here. Thank you. Just a second. Good morning, everyone. I'm a soil ecologist in entomology and plant pathology at the NCSU. Today, I'm going to discuss our work on the interaction between endophytic bacteria and M. macrorhizae. So, plant interacts with bacteria like Microbial and endophytic bacteria and fungi, particularly macrorhizae fungi. So, macrorhizae fungi grow into plants and extend the cell and happy for nutrient uptake. Endophytic bacteria, they grow in the intercellular inside the roots. So M. fungi and endophytic bacteria have overlapped niche inside the roots. So our questions, are they friends, enemies, or friend enemy? So our second question is, do they interact to benefit to their shared host plant? The third question we are asking is that the resources availability affect their interactions. So we use switchgrass as the model systems. So why switchgrasses? Switchgrass is so important. Probably don't already know. So we use two
I'm afraid we've lost Shuijin.
I think we did.
I could give him a call, Mayor and Deborah. Hold on.
Yeah, can you please? Thank you.
Yeah.
We're still a tiny bit ahead of schedule.
Yeah.
So I think we can wait for him to come back in. And otherwise, I know Peter Curdy is on deck and ready to go if we have to switch them up.
Okay.
I hope everyone is planning to stick around for the breakout sessions. You'll notice in the chat that I've been putting the assignments into the chat room. They're also at the bottom of your agenda. And yes, I am trying to fill airtime. So just a quick reminder, Ignacio Carbone and Ben Callahan will be in breakout room number 1. Shuijin,
Deborah, sorry
yes.
to interrupt, but he said that his internet's down, so he wants you to move forward with Peter, and he's going to try to resolve the issue, and I'll try to stay in communication with him to see if he's ready.
Thanks, Mark. That
Thanks,
sounds
Mark.
good.
Okay.
Mary, back to you.
All right, then. So we now have Dr. Peter Ballant-Curdy. Peter, if you're ready, please introduce yourself and go ahead.
Yeah, hi, can you hear me okay?
Yes.
Let's see, I am. Okay.
Peter, we're seeing your presenter view. You might want to switch screens.
Is that better?
Better. Yes, thank you.
Thank you. Okay, well, thanks very much for the opportunity to present today. My name's Peter Ballant-Curdy. I'm a USDA scientist. I'm at NCSU. I'm in the Department of Entomology and Plant Pathology. And this project is a collaboration between me and Kevin Garcia, who's a professor in the Department of Crop and Soil Sciences. I work on maize genetics and pathology, and Kevin works on mycorrhizae. And I think, well, Sweejan, before we lost him, he talked a little bit about mycorrhizae. I don't need to belabor that, but mycorrhizae improve plant nutrition. They improve it by expanding the uptake area, the effective uptake area of the plant roots. They do this by forming very intimate associations with plant roots, and then they have these quite complex transport mechanisms that allow the exchange of nutrients, carbohydrates, and things between the fungus and the plant. The plant gets things like nitrogen, phosphorus, potassium. The fungus mainly gets carbohydrates from the plant. It's a symbiotic relationship requiring a very intimate interaction. And as a plant pathologist, the question I had coming into this was, how do plants resist pathogens, resist microbial pathogens, at the same time as facilitating these intimate connections with microbial symbionts? And how does it do that? Well, I mean, that is not a question that's unique to me. A lot of people have asked that over a number of years. We still don't really have a good answer, but there are a lot of ways the plant does this. But one thing that's becoming clear is plants may have some sort of immunity thermostat where, depending on the stage of growth, depending on the environment, and also depending on their genetics, it may be set high where you have more immunity, so more disease resistance, but less symbiosis, or it may be set lower where there's less immunity, but greater ability to form beneficial symbiotic interactions. The plant can sort of turn this up and down a little bit depending on where it is in its life cycle and what it needs, what its nutritional situation is, etc. Many years ago, we did some work to suggest that the genetics, the maize, you know, the intrinsic genetics of maize lines affects the immunity thermostat. So what I mean by that is we took 300 maize lines, and this figure shows you those maize lines. Each of these spots is one of those lines, and we assessed them for 3 diseases. So gray leaf spot shown on the y-axis, southern leaf blight shown on the x-axis, and northern leaf blight shown colorimetrically here. So the only thing I want you to take away from this is that if a line is more resistant to northern leaf blight, tends to be more resistant to the other 2 diseases. And if a line is more susceptible to northern leaf blight, it tends to be more susceptible to the other 2 diseases. So you have these guys with presumably a high setting in their immunity thermostat, and these guys with a low setting. And if we were to test how well they associated with mycorrhizal fungi, the hypothesis, our hypothesis would be that these guys would interact very well with mycorrhizal fungi, and these guys, which are highly immune to diseases, would also be, unfortunately, also somewhat immune to the mycorrhizal fungi as well. So you have this trade-off between symbiotic and pathogenic interactions. So one of the objectives of our proposal is to test this, and we really have 3 objectives. We want to test this 300-line maize collection for their mycorrhizal colonization and responses to associations with mycorrhizal fungi. This 300-line panel is a mapping panel, so we can actually use that data to then map loci that are associated with variation in the in mycorrhizal association and response to mycorrhizae. And then we also want to work out whether there is indeed a connection between mycorrhizal capacity and disease susceptibility or resistance. And we've started working on this. We have a little bit of money to work on this. We have a master's student at the moment just starting. We've developed a system where where we can do mycorrhizal infections of maize plants in the lab, and then we're extending that now to the greenhouse and to the field. And in the field, we're using this commercial product, Microbial, which is produced by Valent Biosciences. Here, Kevin's group has worked with Microapply in soybeans and shown that it has an effect in soybeans. So that's why we're using it now in our maize work. But this summer, we're applying it to a diverse set of maize lines and trying to see what effects it has in terms of maize growth, maize yield, and also in terms of disease resistance. So the goals and deliverables of this project would be, basically to enable the more effective use of mycorrhizae in maize cultivation. Within that, we want to identify genes, identify genetics, that's lines and alleles that can be deployed with mycorrhizal treatments. And then we also want to characterize the connection between mycorrhizal association and disease resistance so that we can deploy mycorrhizal treatments and associated genetics in such a way that you don't compromise disease resistance. So we'd hope to produce maize lines and maize line mycorrhizal treatment combinations that require reduced fertilizer use, which would reduce costs and associated environmental impacts. Thanks.
Thank you, Peter. And thanks for jumping in.
Oh, no problem.
Are there any questions for Peter, Deborah?
I am not seeing anything in the chat right now.
Okay. All right. Well, I think Shweejin is back online. So Shweejin, are you ready? Is he there?
Yes.
Oh, good.
So sorry for the interruption.
It's okay.
So we have a control, have infected bacteria to 1 and non-1, non-2, and the combinations. So what we found is that after 45 days, what we did, I think I need to go back here. After 45 days, we harvest part of that, and then the remaining part we divided two parts, and half of them let them grow, and remaining half of them will add nitrogen 60 kilogram per hectare. So then we see how they grow, and then what we found is after 45 days, in the bacteria promote mycorrhizal correlation in individually or in combination. But after 90 days, this effect is gone. But when we have added the nitrogen, the effect is still there. And for plant head, this effect is not obvious because actually endobacteria reduce the plant head, so become stronger but less slim. And the thinner numbers a very similar trend with the mycorrhizal correlation. That means when you add nitrogen, the effect become consistent. So plant shoot biomass, a similar trend, and the root biomass also, we see with the nitrogen addition, we see the endophytic bacterial effect be sustained over time. So our work is continue to look at how that the interaction nitrogen affect the input and the interaction affect the microbial or the mycorrhizal community structures. So by sequencing. So coming back to our question, are they friends, enemy, or friend enemies? So yes, they can be friends when they have enough resources. Do they interact to benefit their shared host plant? Yes, as long as the host can effort to support them. So that's carbohydrates. So the resources matters, particular nitrogen. Yes, nitrogen supply created the common currency for both endophytic and mycorrhizal fungi. So with that, the take-home message is the carbohydrates supply from photosynthesis is the key. So With that, I would like to thank Bonnie and Robert who did the field work or lab work, and also my collaborators. Thank you very much.
Thank you, Shuijin. I'm glad you were able to come back and reconnect with us. Are there any questions?
Nothing in the chat, Mary.
Okay. So I think we're ready for our final speaker of the session, Dr. Kelly Zering. If you're ready, please go ahead and share your screen.
Good morning, everybody.
Good morning.
Can you see that? And you can hear me. Okay.
Yes, thank you.
Very good. Well, I'm an economist, and I've given you a little bit of a puzzling title here, so I'll do a little explanation. I'm a professor in agricultural and resource economics, and I'm an extension specialist with quite a bit of experience over a few decades. PhD from Davis in 1984, and I've been here ever since. So learning about agriculture, working with teams of engineers and biologists and veterinarians, and worked a lot on animal systems and more recently plant systems, and getting interested more and more in microbial systems. So this is a great opportunity for me to apply some of the modeling and skills we've built up over the years. So a grand challenge that I wanted to put before you, and something that's driving the work that we're trying to do now, is the forecast for doubling and actually tripling of the value of goods and services produced and consumed annually over the next few decades. This is the OECD forecast. This scale goes from $100 trillion now, 2020, to over 270 trillion by 2070. So in just a few decades, we're doubling and almost tripling the value of goods and services produced and consumed. And that's a tremendous challenge for production systems, and for to do it sustainably is a tremendous challenge for environmental and ecological and resource systems as well. So that's driving us. Let me explain a little bit about this title. This is a fairly recent initiative that we've created. I'm working with people in multiple disciplines to develop this initiative. We're applying design thinking to modeling, analysis, and solutions. I've been teaching entrepreneurship for the last several years, and I've come to understand design thinking as a way to implement human-centric modeling for biological and engineering systems. We start at a biosphere level, so we're looking at a long-term perspective, and so really defining sustainability on a chemical and energetic and biological basis. We integrate all of those dimensions into the modeling we're doing, and ultimately we derive value-based information, and we use it to drive design of specific technologies, processes, supply chains, markets, and policy. So from the very micro to the very macro. In this context, soil is a critical resource and a central process in sustainable production and recycling of food, fiber, energy, and biosphere services. So this topic is particularly compelling for me. What do we do? In this context, we would work with a funder to select the subsystem to be analyzed. So going from the very broad and bringing it to a more specific focus. We would specify the focus, scope, and priority deliverables with the funding parties, and assemble the appropriate co-PIs. So rather than making a specific proposal to you today, I'm talking about how we would work with you to address topics of interest. Generally speaking, we specify the theoretical value model, integrating all those physical, chemical, biological, and human sciences. And we build a corresponding mathematical model, acquire the data, and specify an empirical model. And then we do econometric and other statistical analysis of that data, those data, and employ techno-economic or system simulation modeling to illustrate the combined effects of changes that might arise in the subsystem from a specific technology or practice. So that's sort of the nuts and bolts of what we do. The deliverables from this process, a mathematical model, integrating those physical, biological, and human dimensions into a stochastic value-based framework. So this is where economics comes to bear. And if the funder wishes, we can build an interactive model that the funding parties can use to explore a variety of alternatives. And of course, a written description of the critical interactions in the model and analytical approaches and issues and implications. So what do we do with these results? Well, generally speaking, we work with developers to design a technology to create the maximum value in application. We design and optimize processes in order to make them most economic, most valuable. We design supply chains, so looking at scale and logistics issues, and we design and optimize those. And we go to the scale of markets and actually design markets where they are needed or optimize existing markets, and design policy and optimize that in order to support value being transmitted back through the supply chain to the technology developer. Specific applications may include a feasibility analysis or market opportunity analysis under various deployments of the technology. We may look at key issues in logistics of commercial utilization. So trying to understand how this technology is going to be deployed and distributed, and value analysis, both internal, the profit part of it, and external, being the social part of it, of specific products and practices to enhance soil, plant, microbe performance and sustainability. So that's a very thumbnail sketch of what we do. Be happy to talk to you about specific applications. There's my email if you're interested, and I think I'm in breakout 2. Is that correct, Deborah? I'm sure you'll add that in.
You are in breakout 2. That is correct.
Yeah, thank you, everybody.
Thank you so much, Kelly. Do we have any questions?
I'm not seeing anything in the chat.
Okay. No. All right. Well, I guess that concludes session 2. I really want to thank you all for being here today. I've opened up the breakout rooms, so I think you can just jump in and join the ones you want, and feel free to move in between them. Are you able to see the rooms? Do you see them, Deborah? Oh, yeah, there. If you scroll, yeah.
They're under more.
Yeah, if you scroll all the way down in the breakout rooms in progress window, I see some people already joining in. Okay, so thank you all so much. Deborah and I will be reaching out to some of you and to hopefully discuss partnerships and ways that you can support our projects and faculty and students. We really appreciate that you were here today and participated with us and got to know us a little bit more. So thank you so much. Enjoy the breakout rooms. I will give a 5-minute warning when we're about to close up. So thanks again.
I see Mark
Deborah,
has
do you
his
have
hand.
anything to add?
Well, I see Mark has his hand raised. Mark Cubetta.
Yeah, let me lower it.
Oh.
Hold on. All right. Yeah, I do have a comment. I really wanted to thank Deborah and Mariana for organizing this virtual showcase event. It was outstanding. And to all the speakers for their excellent presentations for the conference. And I also wanted to thank, Ignacio, the director, and members of the center, the CALS administrators for coming, our private industry and government agency partners. And actually there's 5 of the 6 members of our executive advisory committee that are here today. So Vera, Catherine, Cindy, Ross, and Kelsey. So I wanted to wish you fruitful interactions in the breakout rooms. And I just wanted to say that before we went. So thank you for allowing me to say that.
Yeah, thank you, Mark. And Mark, if anyone doesn't know, is our co-director of Cipher in the PSMCC. And we really appreciate his leadership as well. Thank you, Mark.
Thanks, Mark. And Cindy, did I see your hand go up? No, she's shaking her head no. Okay. For those of you that are still here, please go ahead and join one of the breakout rooms. Reminder, Ignacio Carbone and Ben Callahan are in number 1. Shrijin Hu, Peter Ballant-Curty, and Kelly Zaring are in number 2. Johnny, I'm going to say it wrong, Bolidion and Peter Ojiambo are in number 3. Oliver Bars and Wei Shi in number 4. Nicholas Buchler and Nathan Crook are in number 5. And I'll be floating, and everybody, you can always reach me through chat. Chris and Kieran, I noticed that neither of you have chosen a breakout room. Do you need help getting to one?